Hello. Welcome to episode 2. Thank you all guests for being here. We're here to talk about protein crystallography. Yeah. There's a little bit of lack of gender diversity. Just get that out the way very, very early on. We're hoping to, it is important to us here. Thanks to people who've messaged the page so far. We're still, we're always still looking for people to do more episodes, and especially if you're a female and would like to come on the show, give the page a message as well. I think a lot of females underestimate their confidence, so they think, I don't want to come on the show and talk about some things. But, we really want to just hear from people in the field, and it's enough of me ranting about myself and the show. So why don't people introduce themselves?
Great, cool. Hi, I'm Keith. I'm a PhD student in Rutgers University in New Jersey, United States. I did my undergraduate at Montclair State, where I did a lot of dabble in some crystallography stuff that I really became a big fan of, and that's what propelled my interest into kind of pursue more crystallography kind of avenues. So I do a little bit more structural work in neurons now, and some of the proteins involved in the formation of the synapses of these complexes. So yeah, that's what I do.
Hi, I'm Wolfgang, and I studied biochemistry or life sciences at University of Constance, and now I am doing my PhD at University of Hamburg at DESY, the German electron synchrotron. What we are trying to do there is to develop methods to see molecules or see atoms, see materials on a structural level, and I find that fascinating because usually for me chemistry was a black box, but now I can make it visible, and I can also use programming and maths, which I also like, strangely enough, and that's why I'm here.
Hi, I'm Michal. I've done my undergrad back in Dundee in Scotland. Currently, I am a bit further, McGill University, Canada. And my project is very much on a biochemistry of antibiotic resistance enzymes. So we try to see how enzymes are able to be selective on their substrates, as well what are the exact catalytic mechanisms of substrate degradation and how bacteria are able to do it all on a chemical scale. So that's sort of what I'm doing.
And of course, I'm Oliver. I'm the co-host, and I'm a student at the Swiss Polytechnic School in Lausanne, specializing in inorganic and theoretical chemistry.
I haven't had much of a biology background. I've got to put this out there. It'll probably show. And I don't think, Oliver, you either have a big biology background.
No.
So we're challenging ourselves in episode 2 here.
We're on the level of the audience here.
As per episode 1, we're going to start with a dramatic reading of Wikipedia, assuming that we know nothing, which in this case is not too far off. To bring up some nice incognito window here. Were we thinking proteins, or were you thinking?
Let's start with proteins.
Proteins, yeah. Okay, so we're starting the base level here. What is a protein? This is going to be a terrible Wikipedia page, isn't it? It's going to be like 8 kilometers long. Okay, yeah. Oh Lord. Okay. What's not as long as I would have thought. Intro. Proteins are large biomolecules consisting of one or more chain of amino acid residues. They're really just the big, biological molecules, right? They're the. Does anyone have a nice, succinct way of summing up protein? I know what a protein is, but it's sometimes, do you feel ever hard to talking to someone who's doesn't in science at all? They ask, what is a protein? Is it
proteins can be very much thought of as Legos almost. So each Lego has a distinct shape, and we can think about that shape as sort of the chemistry of it. And the sequence of each of the protein is essentially just a mismatch of different chemistries very close to each other. When those are arranged in a very particular conformation, that allows them to perform different biological functions, such as being able to catalyze reactions or being a transport protein, sort of getting things from outside of the cell to the inside. It can also be sort of structural. So having being able to hold parts of organism together. So it's almost everything in your body is somehow related to a protein function.
Yeah, so in this Wikipedia page there's a whole section that says structure, and I suppose that's, as you said, that's the key bit, because all the magic in it, and proteins are real magic in a sense.
Yeah, okay. Maybe start earlier at the at how it's made up. So he said it's made up of Legos. So the Legos in this analogy, these are the amino acids. There are 21 different amino acids, and if you combine them, you can make up. Well, you can combine them in very many ways. If you have lots of amino acids that are like letters, you can make up sentences and many words, many different words. Each sequence typically has a certain structure, but just from the sequence, just from the amino acids that are put together, we usually don't know the sequence yet. So we want to know the structure yet, because, well, this is then what structural biology is about. From the structure, we can oftentimes infer function. So the function of a protein is given or guided by its structure, usually.
Yeah, I think that's great. And I think it's also important to highlight that these structures, when you see a picture of a protein in a textbook or online, it's usually just a static image, and that reflects the environment from which the structure was derived from, that being the crystal. But I think it's also important to highlight that in order to perform a lot of these functions, a lot of these proteins have to be very dynamic in the ways that they move. And there's many factors that go into what makes a protein so flexible. And one being, its enzyme mechanisms. Sometimes enzymes have to move in certain ways to kind of fit in the substrate reactants, so they can make the products and then the release. So it's a very dynamic process. And a lot of the structure elucidation methods that we have currently don't really kind of take in those dynamics too well, but it's X-ray crystallography, in my opinion, is one of the best techniques that we have to get atomic resolution of these living, breathing biomolecules.
Yeah, I mean, but saying that, there is the NMR, which will give you a very good idea of the protein movement. But bottom line is, when we see structures of proteins, they're just a static snapshot of what we're looking at.
Yeah, so all these crystallography techniques are trying to determine how it is in 3D space, because then it tells you how it acts and what its function is, right? Yeah.
Well, that's the idea, anyways.
Yeah, I feel like I'm telling you what your field is. Yeah.
Yeah. Right. So this is the idea, but oftentimes you know the structure and you still don't know what it does.
How is that determined on an experimental level?
So I will be showing you a tiny bit. In my presentation, one way that you can go about it is have a colorimetric assay. So for example, a lot of proteins utilize ATP, which is adenosine triphosphate, and uses the phosphate moiety to somehow change, be it a protein or a small molecule. And we can couple that chemistry to a colorimetric readout. Another thing that I personally use is a liquid chromatography mass spectrometry. So I look at changes of molecular weight of my substrates. So it's a very direct readout. Yeah. There's many ways that you can look at it.
Okay, so I might then jump to, because we're really combining proteins and X-ray crystallography. So I'll then jump to X-ray crystallography. Let's see what this page is like, because this could be a lot of maths.
Oh, yes, it is.
Oh, yeah. Okay, this is very long.
Longer than protein.
Is there a page for protein crystallography?
Oh, there might be. You're right. Actually,
there might be a subsection at least.
Protein.
You can look under protein or macromolecular.
But it's the same principle. So I can give a quick rundown if you like.
Yeah.
One single molecule only interacts very weakly with all kinds of things that we can measure, like photons, electrons. The interaction is very low, and therefore we cannot measure a single molecule, sadly. We're not at the point where we can see a single molecule. That's just what we want essentially. We want to know how this molecule looks like. So there are different ways to go about it. For example, in NMR, you have many molecules in a solution. But in crystallography, what you take advantage of is the crystal itself. Almost all molecules that have a defined structure also form a crystal under the appropriate conditions. So this is just, well, this is not my problem, but it's someone else's problem. And they find the conditions to make the thing crystallize. And then you have lots of molecules that are in the same orientation, and you can shine light on them or something else like electrons, a wave. You can let a wave interact with all of them at once, and then all the interactions add on top of each other. So we now can not only measure the interaction of one molecule with the wave, but all of the interactions with the wave, but they interact in a coherent manner because they are ordered in a crystal. And therefore, we have a large signal from many molecules. And this large signal is, by some maths and so on, related to the amplitude square of the Fourier transform of the electron density of the crystal. So you can show that from the very basic equations, the Maxwell equations. Then you put up a differential equation, and you solve the differential equation, and then you can essentially show how the thing that you measure and the thing that you put in, how they relate to each other. But knowing how they relate to each other, you can then calculate back from what you have measured to what was in the beam, how the electron density must have looked like to produce this kind of diffraction pattern. Of course, this is not that easy. Because this is the whole field, and we can go into depth what the problems are.
I think we've covered everything. Oh, one more thing. So you said the word beamline there. Is everyone talking about synchrotrons, or are we talking about our sources?
Both.
Both. So do we want to just quickly go to the synchrotron page? Because most people won't have a sense of what a synchrotron is. So a synchrotron is just a very powerful X-ray source, right? Do you need more context than that, or?
Yeah, so you have you'll have particles spinning around. I mean, I'm going to do a very disgusting job of simplifying this, but you'll have particles at very high speeds, kind of spinning around this donut, which you see, the classical shape of the synchrotron. And then in order to generate X-rays, they have to be hit off a source, and then the X-rays are generated on the tangent line of that beam line. I'm sure someone else wants to chime in and something that makes a little bit more sense.
Well, it's a bit of a strange thing because you need a really big facility for these. These are like particle accelerators. You can't. Well, I mean, maybe there are some universities. I'm sure there are some universities that have their own sources, but it's a big facility still. That you need to be able to generate these sources. So
usually they are government owned. So, like for example, Canada has only one synchrotron for 40 million people. I think Australia has one as well.
Yep, we got one.
I think Germany has two, or if not more.
Pretty sure we have one in Switzerland at least.
Yeah, and I think you also have X-ray free electron laser in Switzerland.
That's probable.
So we have 3 that I know of, but I certainly don't know of all of them. So I think there are plenty around here.
And there's like 8 in Japan or something like that, isn't there?
I'm going to be wrong
on that. So they need to be that big because the electrons are very fast. So usually in a synchrotron, an actual synchrotron, the particles going around are electrons, and each time an electron changes their speed or direction, it emits radiation. So there's like basic laws of electrophysics. So the larger the ring is, the less energy it loses each time it turns. So you cannot have a very small synchrotron because then it would radiate lots of radiation all of the time. But you want the radiation at the beam lines, and the beam lines is where these electrons are put through a cores. So they are forced through a strong magnetic field, alternating magnetic field, and so they have to change directions all of the time. They feel some acceleration and they change their direction. They radiate radiation, X-rays in this case, because they have to change their course quickly, and they are also fast, and therefore the radiation that they produce is in the X-ray regime. So it is light, but it is very, very energetic light. So each photon has lots of energy.
And Tom, if you want to show the magnitude of some synchrotrons, can you look up the diamond light source? It's called. That's the one based in the UK. It is a beast.
All right. Yeah, and the size of the building is the size of the ring, right? That's the ring that the electrons are going around. Yeah.
I've managed to go there once. It is unbelievable. They have a hotel right next to it for scientists. It's quite a sight.
Countless research labs associated with that synchrotron too that primarily just do structural elucidation work. It's quite a trip.
Huge.
Yeah, I don't want to brag, but the Petra at Daisy has a larger ring. The building is smaller because most of the ring is buried, and the beamlines are just in a small patch of the ring. So you don't need to have a building for the whole ring. You can bury half of the ring or most of the ring, and then only have a building for a small part if you have the space.
Yeah. All right. Well, we might go jump to presentations now. So it's a bit of a long intro, but there's a lot to cover. I mean, for starters, you've got proteins, which is straight out of biology. Then you've got X-ray crystallography, which is straight out of chemistry, and then we've got synchrotrons, which is straight out of physics. You spend a long time in university learning about, all of these aspects. You spend a long time on X-ray crystallography, a lot of time on proteins. There's the whole, there's a lot to know in all of this.
And there's maths and there is informatics.
There's fields within fields within fields, but yeah.
Now, can I share the screen?
Yep.
It has been disabled, so would you be able to enable it?
Yeah, I sure can. Make you co-host for the hopefully that everyone becomes co-host. It's going to be chaotic. Is it working now?
Yes, it is.
Okay.
So
all right, maybe we can all be co-hosts.
Can you see it now?
Yep.
Okay. So hopefully you can all see my presentation. So my name is Michal. To reiterate, I currently work in a center called Centre de Recherche en Biologie Structurale. My French is horrible, so pardon. At McGill University, Montreal. And I will talk rather broadly about what it takes to get to a crystal, since a lot of the audience of this particular channel are chemists. So I think it's important to bring sort of a bit of the base back home. So I work in a lab of Albert Beerhaus. So there's many techniques that we apply in a lab, including X-ray crystallography, nuclear magnetic resonance, and small angle X-ray scattering to look at the structures of proteins, in our case, exactly enzymes. And we also look at the function of those proteins. And to do that, we have to do more enzymatic work. And as I've mentioned before, very briefly, we can use colorimetric assays and LC-MS work. So I'll be talking very briefly about some of them. Anyway, so as this whole talk is about structure of proteins and X-ray crystallography within biochemistry, I think it's important to show how much work it takes to get to that particular point of looking at the pretty structure of a protein. First of all, we have to figure out which protein, which enzyme we want to investigate. And that's more like hypothesis determination. Then we have to get DNA coding for this enzyme. Then the first big hurdle is actually to purify the protein, which in some cases can take months. And in here you'll see the word expression. Expression just means making of protein, then protein purification, confirming of protein activity, crystallization, diffraction, solving phases, creating a model, and then publishing. It is very nice when summarized like that. Unfortunately for me, it took me like 4 years to get to step number 8. So it takes time. Anyway, so protein expression and purification, the major workforce for that is our good old E. coli. So we essentially feed the DNA coding for protein to E. coli, it takes it up, and when a specific chemical signal is given, it starts making the protein. And we can look at whether or not the protein has been made using this particular method you can see on the lower right. It's called SDS page. Bottom line is that this gel separates proteins based on their size. And because we know the size of the protein, because, well, we asked the company to synthesize the gene for us, we have a very good idea of where it will appear on a gel. So in here, we can see that the bacteria absolutely makes the protein. So this, in a well number 1, is all of the proteins from the killed bacteria. And then in well from 5 to 11, that's just me trying to purify the protein. And the protein can be purified in many, many, many ways. Some of them include purifying them based on the size of the protein. Sometimes we do it according to the charge, and sometimes we attach a special tag to the protein. So again, imagination really is the limit. And this is the wonderful machine that we use for, at least in my lab, for purifying the protein. So this is very much a glorified pump. A glorified pump that can read absorbance, which tells us whether or not we have protein, because usually proteins absorb at around 280 nanometers. So that's a very good indicator whether we have protein or not. And in here at the very corner, hopefully you can see a big white column, right? So this is what we call the size exclusion chromatography column, and that very much just separates the protein according to their molecular weight. Those buggers are expensive. One of them can be around 8,000 Canadian dollars. So you definitely don't want to screw that one up. Next, once you have purified proteins, sometimes you want it to be active, sometimes you don't want it to be active, just depending on your experimental design. Personally, I need my protein to be inactive for crystallography, so I have to confirm that it will stay inactive even at very high concentration of the substrate, which is in chemistry, I think, reactants, as well as a high concentration of enzymes, both of which occur in like a crystallization experiment. So yeah, in my case, I use this particular setup of LC-MS. And then comes the dreaded, absolutely dreaded part of getting the crystal. So this can take better part of few years in order to have something that resembles a crystal. Problem is that the proteins are incredibly valuable. So you spend a very long time making it, and you really don't know a priori the condition in which it will crystallize. So we buy commercial screens, which is just a combination of salt, a combination of a buffer at a specific pH, and a combination of precipitant. And you essentially just ute it on the conditions and hope that it will crystallize in a particular chemical sort of environment. But this would be incredibly time consuming to do by person, because those wells are really quite small, and one well has 96 sort of, wells for the mother liquor, which is the combination of the salt, yada, yada. And then we usually have 3 smaller wells for where the protein will be sitting. So essentially, it's 96 times 3 conditions per one experiment. And in my lab, we have this robot, which will be doing that automatically, which is fantastic. It saves you time, it saves you money. And the volumes that we are talking about in this particular case are very, very small. So a big drop would be considered 1 microliter. A small drop would be considered 0.3 microliter, 0.2 microliter. And once you make those wells, those screens, those experiments, the next thing is to actually store it in a very controlled environment. And sometimes crystals like to grow at 4 degrees Celsius, sometimes they like to grow at 22 degrees Celsius. Again, you have no idea a priori where it will crystallize. Anyway, machines like this one, we call crystal hotels, which is really quite a suitable name because all they do is store it for a very long time, and once in a while check up and image the drops in which crystals are hopefully growing. And sometimes those crystals can take a very long time to grow as well, with cases in neighboring labs of mine taking around a year to grow. So again, you don't know. Next thing is, so this is one of my own personal crystals, and it's a relatively big one because it's 0.2 millimeter large, so that's a big one. And they are usually a sign of joy and celebration because you take a very long time to get there. However, the fact that you get a crystal can mean absolutely jack shit, as in they might not even diffract. So sometimes your original hit will not diffract, it will not do anything. So you have to play with chemistry, again, of, well, not chemistry. You essentially just create a matrix of the original screen that you have, in this case, salt, sodium formate, a buffer, and a combination of polyethylene glycol. And you just try to optimize and figure out by changing concentration of, for example, salt and buffer, or the buffer pH, and you will look at where is the crystal getting a better morphology. And once you have a crystal that, well, looks good, the next thing is to actually diffract it. So as a lot of my sort of fellow speakers today, they will say that they are mostly working at synchrotron. Personally, I have access to synchrotron, but it is a few thousand kilometers away in Saskatoon. We prefer to do it in-house. And this is our source. It's gorgeous, it's new, and it actually uses liquid gallium in order to generate X-rays. So yeah, the way it works, you have the X-ray source here, then gets essentially cleaned up to a very specific wavelength. In here you have what we call cryo. So that's going to cool down your crystal to 100 degree Kelvin. And then we have a beam stop. So that will block the direct X-ray beam from hitting the detector, and hopefully we get diffraction. And in here we just have a bit of a zoom in on where the magic happens, the crystallographic magic. You can also see a small microscope here that's just used to make sure that the crystal is exactly in a way of the X-ray beam. If everything goes well, this is what you get. This is an X-ray diffraction pattern. This is almost a holy grail of any crystallographer who has spent countless nights working at trying to get the structure of protein. So there are quite a few pieces of information that one really needs in order to solve the structure of the protein. One is the position of the spot, which is spots, we call those black dots. So they have two informations attached to them that we measure. One is the position of the spot according to the beam, and one is the intensity of the spot. One piece of information that we are really missing is the phase. Phase is important in order to get a structure of the protein solved. In most cases nowadays, because there's such a wealth of structural information of proteins, we can do a little cheat, and calculate the phases based on similar proteins already deposited in our databases. Sometimes it's not possible, but I'm not going to get into that because this is a whole sort of graduate course in itself. So we can reconstruct the phases, and then we get a beautiful protein structure. This one isn't mine, but it's from the lab where I work. And the resolution is mind-blowing into such an extent that we are able to look at even single protons of the structure, which doesn't happen very often, and when it does, it's great, but you have to spend exponentially longer to make sure that all of the hydrogens are pointing exactly where they should be. So it comes at a drawback. So this is a very, very, very brief explanation into what is extract crystallography and sort of how you get to a protein structure. But I do want to take a minute into telling why my lab, well, why lab where I work is really interested in it. So we work on antibiotic resistance enzymes, and it's a huge problem. So now we, of course, have coronavirus, which is a horrible thing, but the amount of people who perished due to this disease doesn't really compare into how bad things might be in around 30 years' time, where current predictions from 2014 are telling us that around 10 million people a year worldwide will die every year due to not being able to be treated for bacterial infections. So in order to defeat the enemy, one really has to understand it first. So we do a lot of basic science into just trying to understand, well, what's the chemistry of this particular enzyme? What are the chemical groups of the protein responsible for disabling the antibiotic? And how exactly is the antibiotic sitting within the protein? Because that's also very important. Using just this information, it can be then fed back to pharmaceutical industry, and they could use it to make antibiotics which don't interact with the resistance enzymes. And that essentially buys us a bit more time. Another thing that the lab where I work is interested in is making what's called an adjuvant, which is essentially a small drug that you add to your antibiotic, and they act in concert to increase the action of antibiotic or prohibit the antibiotic resistance enzyme from working on the antibiotic. So there's many ways that can be done, but this is another thing that we're really interested in. And no scientific sort of talk will be complete without really acknowledging the lab. So yeah, those are the absolute joy of people to work with. So yeah, that's sort of my work in a very, very, very brief summary.
Thank you.
All right, thank you.
Thank you.
Can I ask, so how far into your PhD are you? What's the...
So I am year 5 out of 7.
Year 5 of the 7, right, okay. And that's pretty standard in Canada, 7 years?
It's actually... the standard in a university where I am is around 5 to 6 years. The standard in a lab where I work is 7 years.
Yeah.
Oh, wow. There's a question about protein denaturation, if you have any issues with it when preparing the diffraction measurements.
So, I mean, the protein denaturation, of course, is a problem, but when you have relatively good sort of activity of the protein, you're not really that concerned. So you can do additional work like I've done. It's called circular dichromism. And this is a technique which really just looks at quantity of alpha helices to beta sheets, which are very conserved sort of structural not motifs, but secondary structures within proteins. Usually misfolded proteins are made out of loops and random turns, etc., and they will not have those very highly defined secondary structures. When your protein is folded and not sort of aggregated or somehow else screwed up, they will give a very nice readout in CD, and that's how we get into it. That's how we know that protein isn't denatured.
There was a couple of people in the chat talking about your crystal and saying for some reason it looked delicious to them. Does let me see that crystal one more time? I mean, it's a pretty good photo. Like, it's a good feeling to get that crystal, right? Like,
yeah, but the worst thing is that when you get the crystal and doesn't diffract.
Oh yes.
For around 2 years, I was battling with a crystal that was diffracting to around 7 angstroms. And you can't really say anything about structure at this resolution. So yeah, it was very much just horrible. It was an absolute nightmare to work with because you see a crystal and you know that it will not give you any good information.
How small is it? What's the smallest usable crystal you can have?
This is a really weird question in a way that it's honest. There are technologies nowadays where you can use crystals which really in the very strong microscope to see it. Some beamlines do use microcrystals, but for a sort of person like me who actually has to use their own hands in order to fish out the crystal, I would say that my limit is around 50 to 25 microns.
Still not.
So we've measured crystals that are way smaller than that, around 200 nanometers.
Yeah. It's tiny.
Yeah.
It's not decent.
How do you even how do you even work with that?
The smallest ones are just in a solution, and you shine light onto the solution, and you hope you hit only one. And sometimes you hit 2, and sometimes you hit 3, whatever. But yeah, and then because you need so high radiation, one shot kills them. So you can have one picture from one crystal.
Oh,
but it's
God.
like looking at like making an X-ray projection image from a person. You don't have 3-dimensional structural information in a 1-dimensional in a 2-dimensional picture of the thing. So you need many 2-dimensional projections to reconstruct the 3-dimensional thing.
Mm-hmm.
So you need to be able to repeat this and hit these very small crystals very often. And well, yes, it can be done.
Like a pain.
Yeah.
There's someone asking about high field NMR before we move on.
I don't touch that with a 10-foot stick. Those NMR is not really a thing that I very much use because I don't want to play with it. However, we do have some, and I know that it's possible to solve the structures of proteins using NMR. Right now, the main limit is the size, because the bigger the protein, the more peaks you have, and the worse the resolution gets.
The building blocks of a protein are always the same. They are amino acids, so they all have peaks in a similar range, and so they necessarily start overlapping. You have a 3-dimensional structure, but you have only a 2-dimensional measurement. Therefore, it doesn't scale well with the size. But you can already find remarkably large proteins, or solve the structure of remarkably large proteins with NMR.
All right, shall we jump to Keith's presentation? I think I got to make you co-host as well.
Oh, all right, let's do it. I'm ready.
Please don't kick me.
I'm the only one who can co-host now.
Yeah.
Does any of this
make
sense?
And I see that someone asked if I ever dropped a protein crystal.
Oh, yeah.
Yeah, I have.
Yikes on bikes. Anyway, hi, my name's Keith, and I did my undergraduate at Montclair State University in New Jersey, where I dabbled in some kind of crystallography projects. So what I'm going to talk to you about today is what I did for my undergraduate, which was the structural elucidation of an enzyme called dihydrofolate reductase from an organism called Wuchereria bancrofti. So what is Wuchereria bancrofti? Well, Wuchereria bancrofti is one of the 3 primary causative filarial nematodes that cause lymphatic filariasis. And why that's a problem, you ask, is because the symptoms are elephantiasis, and you have these gigantic limb swellings and genital swellings, and general not having a good time with elephantiasis, right? So the goal of this kind of project was trying to determine if we can use this enzyme and determine structural information from it and see if we can make an inhibitor specific against the dihydrofolate from the Wuchereria bancrofti organism. You feel me? So dihydrofolate, what is it? It catalyzes in a very important mechanism from dihydrofolate to tetrahydrofolate, which is an intermediary in a plethora of interactions that have to do with involved with the synthesizing of amino acids, some amino acids synthesizing some of the bases in DNA. So it's a very, very attractive drug target for a lot of these parasites and bacteria. If you have an ear infection or a UTI, you will more than likely be giving a DHFR inhibitor to kind of quell that infection. So it's a very well, oh, my cat's right here, actually. Hey, how's it going? So it's a very, very important enzyme. So currently, there are no structures of dihydrofolate reductase in the protein data bank or just of any filarial nematodes. So that's the kind of novelty within this kind of project. So again, dihydrofolate reductase, a potential drug target. This is a little bit of chemistry because I know you chemistry nerds really want to get into that. This is just showing that on the upper right up here, oh, you can't even see my mouse, but on the upper right, you start with folic acid or folate, which is usually obtained through your diet or through any other means. Which is then reduced to dihydrofolate and then to tetrahydrofolate. And you can see that the right aromatic ring of that pteridine ring in folic acid is actually getting reduced, which has its own physiological implications. And you can see that tetrahydrofolate on the right is involved with, just so much stuff that is involved with the catabolism of a lot of these building blocks to sustain life. And my cat is going to say hi. Hey, how's it going? Woo! Oh. Hi, it's my cat. Hey, Damon, say hi. Anyway, today there are no structures of the HFR, which make this a very interesting project. So let's just keep going. Hey, how's it going? Okay, so previous inhibitory data from our lab has elucidated some of the compounds that actually work well or inhibit DHFR. One being methotrexate, which is a folate analog. So you can imagine that it's an inhibitor that looks very much like the substrate. So it's going to bind competitively to the active site of that enzyme to kind of knock it out or inhibit its function for a lot of reasons. So these right here, what you're looking at are the Dixon curves. These verify that these inhibitors that we are actually very well established are actually they're actually competitive inhibitors through DHFR. So moving on, Michal did a great job in terms of touching on the purification of a lot of these proteins, and the purification of every protein can be different. Luckily, oh my goodness, my cat is stepping on my keyboard. Okay, I love you. Whee! Okay, anyway, we're gonna just getting back. I already spoiled a lot of this for you. Anyway, let's just, all right. So the purification method that I had used, didn't have any access to any FPLC system at the time. So a lot of the purification protocols involve using these fast protein liquid chromatography to, with that glorified pump to kind of pump the protein through the size exclusion column to kind of separate these kind of proteins by weight. And that's how you kind of get a more pure fraction from your original purification protocol. So with this, I had to come up with something a little bit different. However, you can actually, if you can buy agarose covalently bound resin to methotrexate. So that in itself can serve as a purification resin because it's just this precipitate material that has methotrexate, which is a competitive inhibitor for dihydrofolate. So in theory, we can actually use that to purify it out. So basically the steps of this purification involves we have to grow up the protein, express it in our bacteria, E. coli. I should say that it's a very non-hazardous strain of E. coli, obviously. We take that, we induce the expression of it through a T7 system, and then the bacteria grow it up, and then we lyse them, and then we first subject it to a methotrexate column. And what will happen is that those DHFR molecules, along with the endogenous DHFR molecules, mind you, will bind to that methotrexate resin. So once they're bound to that methotrexate resin, then we have to get them off somehow. So how you do that is you have a buffer that has a higher concentration of something that looks like methotrexate, like folic acid, which is cheap and abundant, and you can buy it in kilograms. So what you could do is you could pour the folic acid over this methotrexate bound DHFR resin, and then it will elute from the resin, giving you a more pure product from which you started from the lysate. And I could take that methotrexate agarose eluate and put that over a nickel affinity column because this DHFR construct has a hexahistidine tag, which allows it to be purified over a nickel resin. And that's a whole affinity chromatography can be videos in and of itself. But once I have it eluted from the nickel column and we use imidazole, which has a very high structural similarity to histidine, which allows us to elute it from that column. And then we have this pool of pure protein, but it has folic acid, probably. It has a bunch of imidazole, which is, you don't want high amounts of imidazole in your protein solutions because it is acidic. So I have to dialyze it out, and that's just simply, I put my protein in a tube that is porous, and then I put it in a beaker overnight, spin it around so I can diffuse some of that, some contaminants I don't want out of my purification prep. And then after that, I spin it down, I concentrate it a lot. You need, for the smaller sized proteins, you need to kind of concentrate them. They have to be at a high, like milligram per ml concentration in order for you to begin your crystallization trials. So once I had all that, this is a spectrograph, I think, an absorbance spectrum, I'm so sorry, showing the different kind of absorbance peaks that we see. So on this, we see that we see 2 peaks at around 230, which is just a general catch-all. I believe that's the peptide bomb that is absorbing. And then we also see a high peak at 280, which if you are able to know the extinction coefficient, if you remember Beer's law, for your protein, which can be calculated online, and you know the molecular weight, you can fairly precisely, with a good amount of accuracy, determine the amount of protein you have in a solution, which is very, very important when you're doing these crystallization trials. So this is another SDS page to demonstrate how successful our purification protocol was. The first line, so this SDS page will separate proteins on a mass charge ratio. So you'll see that the higher molecular weight proteins stay up top and the lower ones just kind of go down bottom. So in the first kind of row, we have the lysate. Following that is a flow through from a column. And then we wash it, we elute it in lane 4. You can see it's still not pure enough yet. So we have to purify it up a little bit more. And that's where the methotrexate column comes in. So we put it over the column. We wash it a bunch with a buffer that doesn't contain really much of anything, usually high salt, because a lot of the contaminant proteins will bind to whatever agarose system you kind of have as your purification resin. Then you elute it, and then, oh my goodness, you have lane 8, 9 with pure protein that you begin your purification trials. Usually what I would do after this is I would determine the enzyme activity just to make sure it's as what I would expect it is, because you don't want to be doing, you don't want to set crystallization trays. If you don't have a robot, you don't want to be sending crystallization trays with protein that may be funk. So after years of nothing, and I want to emphasize this nothing, there was nothing in these crystallization trays after so long, and after, whatever. And what I ultimately had done is, since it is an enzyme, usually if you add usually already known competitive inhibitors along with whatever cofactor it needs, it has a way of kind of locking up the system, so it's a little bit less flexible and it's more likely to nucleate and form crystals. So I added the cofactor for DHFR, which is NADPH, in these crystallization screens, which have a wide amount of conditions, usually most popular being ammonium sulfate and PEG. So I'd added the cofactor to this crystallization solution. And that was able to form, as we'll see here, crystals after so long of not seeing a darn thing. And the top 2 pictures are actually crystals of ones that reflect the ones that we used to actually use at the synchrotron. So the bottom row of kind of crystals, they were kind of like, trials. Actually, the bottom middle one is actually the first crystals I actually saw. The right one is some crystals of that same exact solution with an additive. Usually if you have like needle crystals, a lot of synchrotrons now can shoot crystals that have really strange morphologies. And so that's not really that much of a problem, but usually you can buy kits that have additives to add to your crystallization solutions that will hopefully help it precipitate out better. And the one in the bottom right is some myoinositol additive compound, which was able to change the shape of the crystals from the bottom left to the bottom right. So that's the importance of additives. So this is again just showing we purified it great with no fancy high-tech pumps or anything, just pure gravity and resin and sigma. And we were able to get those crystals, which is pretty good. They were about roughly 100 microns in size, which is not terrible. And I found out that if you kind of adjust the pH in a little bit range, you can get some dimensions longer than others. But those reflect best the crystals that we shot. So they're kind of pretty, but do they diffract? So what we had ultimately done is that my PI had made some connections with this very nice woman, Fairleigh Dickinson, who was able to allow us to piggyback some of her crystals that she was diffracting. She was able to shoot some of our crystals kind of for us. So they're pretty, but do they diffract? So this is kind of, this is at Brookhaven National Laboratory. And you can see on the right panel, that's the infamous donut shape. In the middle, that represents a diffraction pattern of reverse transcriptase, which is the enzyme that is able to kind of violate the central dogma or whatever when you're able to convert RNA back into DNA. That's a very popular enzyme that is involved in HIV, which is a very big, obviously, something that you want structural information for so you can design drugs for. And on the left is actually the beam line. It's not very informative in terms of what's happening here, but there is, you can see the robotic arm that's able to kind of pick up these crystal things, these little loops that you have to load the crystals onto, able to pick them up and position them in just the right way and twine them up with the X-ray generator. So I actually went to the Brookhaven National Lab on April 29th, 2019, which was my 23rd birthday. So that was like good stuff. So do they diffract? Hell yeah, they diffract because I made them. They diffracted to about 2.7 angstroms, which is, not, it's not amazing, but it's definitely a good amount that you could actually get information for if you want to find a structural justification for whatever phenomena that you're seeing. So 2.7 angstroms, not too bad. So then again, we have just more pretty pictures of crystals and does a diffraction pattern. However, does the work end now? No, it does not. It has just begun. You have to build that model. You have to refine that model. You have to make sure every single angle meets up with Ramachandran guidelines and all these different things. You have to really refine this structure. You have to build it up in a different program. And it's the most fun part of the entire process, in my opinion, is when you you've solved the actual density issue. And one way that you can do that, as Michael was mentioned, is through something called molecular replacement, which is exactly if we have the structures of known proteins, we can kind of use that information to kind of infer the phases. However, there's a lot of problems with that. I could tell you, I'm thinking of a spy who is charming, who is funny, and is very charismatic. You might be thinking of James Bond when I'm thinking of Austin Powers, and that's the kind of bias that's kind of evident in this kind of system. So there's a lot of, and that's why there's a lot of bias from previous models that can kind of propagate through different crystal structures that rely on a certain protein structure to build off their molecular replacement to establish the electron density. I just said a lot of stuff. Don't have to worry about it. It's a lot of math. It's very complicated. But the fun part is that when you get to fit those kind of the bonds and the atoms inside the electron density, that is the most fun part. You usually go through the sequence of the protein and you try one by one shape these amino acids into the density. And there's a few programs that can do that automatically, but it's really fun to do it by hand, if I must say. And this is some of the data that comes out of this refinement. So this is the resolution range. It tells you the space group, which I really won't go into, the unit cell dimensions in angstroms, and the two important values here, which I won't really go into, are the R work and the R free. Those kind of really determine how good your refinement process is. And these are just some more details. The amount of Ramachandran favored acids, side chains, or I'm sorry. My goodness, I'm hitting my mic. There we go. It tells you the percentage of the amino acid angles that kind of fit into the accepted, how they, the Ramachandran outliers tell you if the angles between your peptide bonds are where they should be and they don't violate any kind of known phenomenon. So interestingly enough, I had set these crystals just with, in my mind, DHFR and NADPH its cofactor. So in my mind, I'm thinking, okay, this structure is more than likely just going to have NADPH and DHFR because I had dialyzed out all of the presumable folic acid or imidazole. However, assuming is a very dangerous thing, as we'll find out, because when I was solving the structure, when this structure was being solved, we noticed that there is a kind of a big blob in the active site, and then it became very apparent to us that this is probably the folic acid that we used in the purification. So this folic acid, if it weren't for the purification protocol that we were using to kind of purify this protein out, we may not have crystallized it in the way that we did, because they more than likely crystallized because they were locked up because they had that folic acid still bound from the initial stage of the purification. So that was a very serendipitous event that we were able to kind of take use of. And also you can actually kind of see that the density for the folic acid isn't as strong as the electron density for a lot of the other side chains. And that's probably because it's probably dissociating. It could be even reacting. And there's a bunch of reasons why the electron density for the folic acid in this case is not as strong as, say, NADPH, the other cofactor. So I'm going to show this little movie. Movie was made in PyMol. It's going to go over the general shape of the structure. I want to say that before this, the woman that helped me kind of walk me through the structure, because this is not something that you can really just do and learn in just a few weeks. It's a very long process. It's very confusing, and you have to learn quite a bit, as I'm sure a lot of you have. But what one group did is they used molecular replacement from one model, and I took another model to solve for the phases. So we took two individual phasing models, and we both were able to make the same structure. We're able to both build the similar structure from these two different models that we had to use to get the the phases of the diffracted X-rays from. So this is just a little video. It's going to rotate this. I'll touch on what that red part means in a little bit, but this is the active site of DHFR. Now it's zooming into the folic acid in its active site right next to it using glutamine. I believe that's 32. That's mainly binding the folate. And now we're going to focus on, that's the nicotinamide ring. That's the active part of NADPH in this reaction. And we can see that there's a bunch of waters that kind of, those little spheres that you're seeing are actually waters that kind of make it into the structure. And you'll see, if you ever look at electron density data, you'll see a lot of like little blobs that, since usually we can't detect hydrogens in diffraction, they'll just be single points of just blobs, which are presumably a lot of times to be water or salt or what have you. And this movie was generated through a program called PyMol. It's free for academics. You can look up any protein on PDB and just look at its structure and kind of have fun with it and see what it's doing. So that's a very a neat thing. Some structural characteristics of it. It has the standard amount of alpha helices and beta sheets that you would expect. There is in the original sequence for this protein, there was an omission of a very important amino acids that we had to substitute in the actual construct of this protein in order to get it to work. So what those red things that you were seeing earlier really reflect the omitted amino acids from the original UniProt sequence deposition. And I have one of the people with whom I worked, Dr. Uli Gubler. He is a big fan of 3D printing, and he made me one of the 3D printed models of actually my structure. And that red little thing right here reflects the amino acids that I was just talking about. And it's really kind of a neat thing that I very much cherish. Once I had seen that, we have folic acid in our structure, can I see that like in my enzyme prep? Can I just take the prep out of my stock and see if the KCAT is sufficiently different still? And it turns out that it is. So we see that folic acid and methotrexate both inhibit the actual turnover rate of this enzyme, opposed to just eluting it with imidazole. So there is folic acid presumably still bound in the stock of this concentrated protein. And this is just showing, this is some other data of a methionine that isn't there on the active site that we could potentially use as a source to kind of potentially draw drug interactions with. And this is just overlapping some DHFR structures. Since Butyrate bancrofti is a filarial nematode, it puts it in a weird evolutionary tree between bacteria and eukaryotes. So we kind of wanted to compare what's happening there, and it turns out that a lot of these DHFR molecules are structurally conserved, but they're not sequentially conserved, which is a very interesting thing, because if you think, the structures are derived from amino acids, obviously, well, that infers that there's so much more happening in terms of what's making these structures fold. And we still don't know, obviously, a lot of the details in terms of the exact physics. I'm sure some of you have heard of the Leventhal's paradox, where how could a protein search through all these perfect conformations and adapt its native structure? It would just take way too long, but that could be its own phenomena. But I had pretty much finished with my spiel, my blant, my whatever. I'll take questions if anyone has any.
Thank you. Have you ever dropped any crystals?
Drop plates. So the hotel that Michal was talking about, my hotel was inside of a styrofoam box in the cold room. That was my plate hotel. So it really was just one firm kick away from just ruining my entire stuff. But I have dropped plates. I was in my first rotation of my PhD. I had dropped a plate in some crystallography lab. It was just Oh, I've done so much things. I've dropped a beaker. Oh, so much stuff.
Happens to everyone. Someone's asking, how does the crystallized protein vary from the one found in your body?
Oh, that's a great question. There are a few structural differences that we see. I could actually just open that PyMol right now and show you if you like. So this is WBDHFR. There are a few amino acids right around here that are responsible for pinning down this adenine binding pocket. So this right here is adenine, the adenine ring, adenosine ring, whatever. In the human forms, there is an amino acid arginine that comes over and reaches above top of this adenosine moiety, which in this structure is lost and it's not found. However, in its place, we can actually, I don't know which water it is right now, but a few of these waters actually go above the adenosine ring, and they form water bridges with other amino acids. So that kind of in and of itself, you're having like a water bridge between amino acids that are fairly far apart in order to keep that adenine ring down. So that's one of the interesting things that we found.
Does that lead to a functional difference?
It does not actually. That's still kind of being looked at, but in terms of KDs, not too much. But there's a few differences in the structure that are just kind of interesting and may have that deviate from humans or E. coli DHFR.
I think we'll push on to the third presentation, and we'll come back with some more questions at the end if anyone has any questions from chat.
Yeah, nice. Okay. So that's me. Why I'm in crystallography is I want to see molecules. I want to know how they look like, and I want to know how they work. Proteins are just one kind of very large molecules. And oftentimes when we do chemistry, it's like a black box. But I'm a visual guy. I want to see what is going on. And I like chemistry, but I like it even more if I can see something. And also I want to know how drugs work, or that's how this obsession started. And usually, and I think the chat will agree, when you go into chemistry, it's either drugs or explosives, and maybe both. And so this is one way to find out how drugs work. And I also have always been torn between informatics and chemistry. This is what I can also do here where I am. So I work in this very large group with very many nice colleagues. We're all guided by this genius in the middle. This is Professor Henry Chapman, and he comes up with all these nice ideas how to use coherent radiation of all kinds, like electrons, neutrons, X-rays, to look at matter on an atomic level. So there is X-ray microscopy, there is X-ray diffraction, there is electron diffraction, and many other things. I usually in the crystallography part, but I'm helping with the other things as well. So his ultimate vision, and I think he needs to do that before he can die in peace, is to see a single molecule, because he doesn't really like the crystals. But sadly, you would need around a 1 with 14 zeros, photons, to see a molecule directly. So that's a lot of photons. And we are working on it, I promise. And you might have heard about the European X-FEL. We would only need to make a 10,000 times larger machine, or make this machine 10,000 times better, or a combination thereof. Maybe someday. It already cost in the billions, I think, to build this machine. So, okay, until we have that, we need many identical molecules, and they need to be oriented. Because if they are not oriented, then you get a radially smeared picture, because they are in all potential orientations. What you get would be a radially smeared something with not much contrast. You cannot see that much. You can basically estimate the rough size, but then that's it. So you basically have one-dimensional information in that case, but you want to determine something three-dimensional, so this will fail very quickly. But if they are oriented, for example, in a crystal, but also any other means that you could use to orient molecules, but there are not that many. So usually this is a crystal. Then you can measure many molecules at once and get a strong signal from them, even if you don't have that many photons. Almost everything crystallizes. This is the nice thing. Well, if you find the right conditions, and if the thing that you want to crystallize has a defined shape. If it doesn't have a defined shape, it cannot form a defined crystal. But if it does, then it crystallizes. And in the lab, in a regular chemistry lab, for example, sugar, salt, all the things crystallize. And everything that crystallizes, you can put in a beam, and you can measure the diffraction, and then you can determine the structure. So this is a very nice tool, not just for proteins. It is basically every chemistry lab, in my opinion, should have some home source, because the smaller molecules make even nicer, larger, better diffracting crystals. So it's basically settled science for the small molecules. But you still get very nice and very good-looking structures. So this is the European X-FEL. The European X-FEL is like a synchrotron, but in a straight line. So you can accelerate the electrons even more, you make them even faster, and then you milk their energy even more with all of these undulators. So they have to go through a alternating current, alternating magnetic field, and this magnetic field is alternating in so long and so dense and so strong that the photon flux within this undulator is so high that it starts lasing by itself, and thereby amplifying the amount of photons that come out. And thereby you get very high amount of photons, around 10 to the 10 photons. So this is what crystals look like. For example, this were now the main protease from coronavirus crystals. So that's what we're working on right now, or this is one of the projects that our group is working on right now. I can't show you what the results are from that, but we'll publish soon. So this is one of the beam lines. So where we would put the crystals in, around here. Then we have a detector that's there. Then there is a robot arm that puts the crystals into the beam. Well, lots of instruments. It's very chaotic, and it always looks slightly different each beam time, of course. A beam line, of course. Oh, this is just some beautiful image. This is how a diffraction pattern would look like if you put ascorbic acid powder into the beam. So this is what I did. So to calibrate the detector, because you need to know the geometry of the detector, and if you don't know the geometry of the detector, how these two panels are arranged, then you need some standard. And this, as a standard, we just used ascorbic acid. It's not the best idea, but this is all that we had at that moment, and it looks beautiful. That's why it's here. This is a diffraction pattern of a small molecule in an electron beam. So this is the actual direct beam, this very bright spot here. And then because crystals are arranged periodically in all three dimensions of space, the diffraction pattern has peaks. This is to do with the Fourier transform. You can see the peaks here, and this is basically to show you can do the same kind of experiment with all kinds of waves. It doesn't need to be photons. It can also be electron waves. And this would then look like this. And this is some diffraction pattern. The white spots are the actual measured data. And the orange brackets are where we would expect to measure the data, the peaks. And this is where I come in. Of course, we already know where we expect them. Otherwise, we couldn't do the experiment, and otherwise we couldn't measure the intensity here. But I'm there to help to make this better, this part, to know better, and for all kinds of experiments. For example, this is a different kind of experiment where you don't clean up the energy of the beam, but you use just the beam as it comes directly from the undulator. Usually, you pick out a specific wavelength and do the experiment just with this light of this one wavelength or of this color. Of course, you can't see the color, but it's like only taking blue light and not all of the other light. But in this experiment, it would be taking all light, like white light. And then the prediction becomes more difficult, and then I come in and help. What you end up with when you do all these things in the end, you get some electron density. This is these blue mesh here. This is what you actually measure. And then within this blue mesh, you can try to interpret this blue mesh and think what kind of atoms fit within this blue mesh. And this can be very useful because in this case, this is an old experiment, not from our group, but we're actually doing the same thing basically right now with the main protease of coronavirus 2. And what you're seeing here is a part of the electron density of the main protease of coronavirus 1. And there is some inhibitor bound to the protein. So not all of the protein is in this ball and stick model, but in a cartoon. But of course, it's still made up of atoms. It's not something special. A protein is still a molecule. So my work is usually lots of maths, integrals, derivatives, statistics, lots of programming, and helping out wherever I can, also data processing. And what I'm working on right now is to make this prediction, what you can see on the left, fit to what you measure, what you can see on the right, as perfectly as possible. So I want to have basically every pixel to be perfect. That's the ultimate goal. I also wrote software which is XDS GUI, which maybe some of you have used already. This is a graphical user interface to use XDS, which is one of the programs that is being used. I'm also collaborating with Crystallography. This is the software that is developed with our groups, mainly under Thomas White. And yeah, so XDS is also one of the softwares that I'm helping out with. And in my spare time, I develop hash tables. I love them. Good.
Thank you.
Cool. Thank you.
There's some questions about why the pictures look so beautiful. Where do the colors come from?
It's just in reality it's just intensities. So it's just a value between 0 and 1 maybe, just some relative intensity. But I just choose the color scale so that it looks beautiful.
It's smart,
so,
yeah.
But
yeah, right.
really very nice.
So this pink is some intermediate value, this blue is some low value, and the green is even lower, and the black is 0, and the white is the highest most value that you can have. And here there's a different color scale. So here the red is basically the saturation value, the white is an intermediate value, and the blue is the 0.
What is the efficiency of the cysteine linkages in your suicide inhibitor experiments? Because I saw...
This is a very good question, because this hits exactly where why we haven't published yet, because we don't really know that.
Yeah. Oh, good. That's
yes.
really cool.
So we're looking at that. We think the efficiency is of the things that we found is very low.
And that's seen by the density, right?
No, so the density is very good for what we have, but we used very much of this stuff. So the efficiency could also, or maybe I'm not understanding it correctly, but what I think is the efficiency might not be good, but if you use lots of it, then you still have a good density in the crystal.
Wolfgang, your camera's off, I think.
Sorry. Because of the screen share, I think.
That's all right.
One quick question: Were you drinking from an Erlenmeyer earlier, a flask?
Yeah, I know. I know you're not supposed to do that, but I promise that they have not been used for anything else but drinking from. And they're cheap, $3.50, and borosilicate.
Interesting. A question about, so you're saying you're giving number of photons like 10 to the 10 photons. It's bright. It's like, it's obviously, but like compared to like, sunlight or something, it's still like billions of times brighter than that, right? Like what's a scale?
Well, it
what's a, yeah, what's a
per area? Yes, per area, yes, but not in general. If you
of course,
yeah,
the sun is huge.
yeah. So if I look into the sun and then I look into your X-ray beam, which is not advised, but like your beam is like, what would you say, like millions of times brighter, like thousands?
Oh no, maybe thousands.
Maybe thousands, yeah. But it's just looking at number of photons. It's hard to grasp, how bright these things are.
Yeah, but you also need to weigh up each photon has a different amount of energy. So the photons in the X-rays have much more. They have 1,000 times more energy per photon, but they also penetrate deeper. So it's a bit hard to weigh them up. Basically, what would happen is you would get a very tiny column of damage in your eye.
Also talking about the sort of brightness of the beam, there was an accident a few decades ago in the Russian synchrotron where I think he was a technician or something like that. He was responsible for the maintenance of the beamline, and he looked directly into it and then turned on.
Oh, God.
So he had essentially a sunburn going through his brain.
Basically from one side to the other.
Oh, yes. God.
Yep. And there are actually, I think it's an MRI or something like that. There are images of the damage. He lives, but he unfortunately has seizures and some other problems now. Thanks to that.
It's horrible. Okay,
so
there's
don't look into the X-ray beam.
You mentioned being able to work with very small crystals in solution. How do you orient the crystals?
It doesn't matter. So hopefully, actually, they are not oriented. Because if they are not oriented, then we see them. If we see them oftentimes, we see them from all different orientations, and that's what we need to do. Of course, if we had some way of rotating them, that would be even better. But we just hope that by chance we see all of the rotations, and then we later find out what the rotations were. And this is also part of what I'm doing, because if you have a random orientation and you have just a single picture of a random orientation and you need to find out this orientation, well, this is hard, and this is called indexing, finding this rotation. But it can be done.
Okay. What does a unit cell of a protein crystal look like? How large is it, and how do you determine it?
Typically about 10 nanometers in all directions, but of course, much more, much less. It at least needs to be as big as the protein itself, the molecule itself that you're measuring.
How large can you go when it comes to? Can you crystallize like whole viruses out, for example?
Yes, and we have measured them as well. And some viruses crystallize.
All right.
It's amazing.
Okay. All right. Well, thanks heaps for the presentation. I might just jump to some general questions, get everyone involved again. Because for me, So I'm not in this field, obviously, as you can tell. What do you think of the field? And like, do you recommend it to others? Because just coming from an outsider, you've got these kind of two very distinct sort of, I don't know what the word is, but on the one hand, you can really attack these sort of real world problems, and it feels like you're curing diseases in a sense, or that you can kind of approach that way. You can say, oh, I'm actually it feels like you're helping people. But on the other hand, a lot of people feel like they're just beating their head against a wall for years, waiting for things to crystallize. I mean, you're doing stuff as well. So it seems like, did you get what I mean by that?
Yeah.
Like there's these two kind of, like quite distinct sort of sides to it. Like what? So anyway, I'll stop talking.
I would say it depends on how much you hate yourself. If you really hate yourself, you can enjoy crystallography. But in all honesty, there's a lot of nuance to that particular question, because some people would say that crystallography is slowly a dying field, but people have been saying that for a long time already. So it's still a gold standard for elucidation of protein structure. And as someone I think previously said, protein structure essentially means protein function. So this is still the best way of figuring out how a protein looks, how it's able to create, how it's able to exert a chemical reaction, and in quite a few ways, this is also giving us information on maybe how we want to stop this chemical reaction. It's honestly a great field. I've learned a lot. I'm from much more biology background with a bit of biochemistry, so I had to learn a hell of a lot in a short time. But it's genuinely a very enjoyable ride when it works. When it doesn't work, there are so many variables as to why it might not be working, and it's next to impossible to vary them all. So if you're a stubborn person, great. If you give up easily, try something else.
Yeah, I don't think there's anything more convincing than an image, to be honest with you, and to be able to see what you've been working on for 2 or 3 or 4 or however long trying to get a crystal is so satisfying. The kind of aha moments in crystallography and structural resolution are far and few between compared to, I guess, normal quotidian research. But it is so satisfying to be able to see something that you've been just working on, and for the first time, maybe even. And I think it's just an absolute thrill. And I think if you're curious about chemistry or biology and even physics, it's the holy trifecta if you want to get into crystallography. 10 out of 10.
Yeah, I'm not doing the crystallization myself, and I would not recommend that unless you have a very long breath. But all the other parts about crystallography or structural biology are great. So ideally, someone else that gives you a crystal. But even if not, the great thing about this field is that you can combine so many different sources of knowledge. On the one hand, you're forced to learn about physics, maths, informatics, chemistry, but on the other hand, you get to use all of these different things and have a lot of variety. And if you do it right, in the end, you have a general knowledge of all of these things. Well, of course, not of all of physics, not all of chemistry, but yeah, a more general than you might have when you do just some biological experiment.
Do you have any advice? Oh, sorry, cutting in. Do you have any advice for people looking to get into this sort of field, or say they're just starting university and they're thinking, biochem, do you have any advice for people in that situation?
I think a lot of research places, universities that do research or colleges usually will have people who do some sort of structural elucidation, whether it be through NMR, CurrentChem, or crystallography. I think that's really the best way to kind of get into it. And if you're curious about the math, there is definitely a lack of people, myself being included, who really don't have a mathematical fundamental physical understanding of the properties of X-rays, diffraction, and calculating Fourier transforms by hand and stuff. But there are so many resources, and even people at Synchrotrons are actually willing to help you. Some people at Brookhaven actually do like online classes and stuff and things like that for people who are interested in crystallography. So there's resources are bountiful, especially for this.
Do you guys do much coding? Would you recommend people sort of look at, because when you think, oh, I'm going into biology, a lot of people I know biology do pretty heavily with coding and computers. Would you recommend that's something, if someone's looking to biology, and is that a necessary evil for you, or is it something you enjoy?
So it depends. For example, I don't have to do any coding at all. My knowledge of coding is that I know that Python exists, and that's about as far as it extends. So at least as someone who's, in crystallography from the start to the end, I don't have to touch coding. I might have to install things using command line, but that's it. But it depends sort of, again, what sort of field of biology you want to go to. If you want to go into sort of bioinformatics or sort of population biology, then yeah, coding is a necessary evil. Biochemistry, not necessarily.
I think Wolfgang definitely enjoys it with all the stuff.
Yeah, exactly.
I do.
Yeah.
So I almost exclusively do coding. So my PhD is about writing programs and processing data, and I help out where I need to, but yeah, mainly I do coding. What I want to say is that what you usually learn in bioinformatics is a different kind of informatics, or different kinds of techniques. So you learn about sequence data, and you learn about alignment of sequences, these kinds of things. But in crystallography, we have more images and more structured, actually. The data is more structured. So you have either two-dimensional images, or you have three-dimensional densities, or you have the actual structures, which are connected atoms. So that these are completely different kinds of data. So even if you feel well equipped in bioinformatics, you go into in crystallography, and you might not recognize many things. So if you plan to do that, you should just in general try to learn informatics. It's not that hard, in my opinion, but it's not that easy, because if you say bioinformatics and then you come into crystallography, that's what I want to say, then you might still be just as well off as someone that knows just informatics and not bioinformatics.
So no regrets about the field. Is that we're all
not from me?
No. Yeah. Oh, it's good to see. Wow, it's sheepish.
What resources would you guys recommend to people who are interested in the field, like books or online resources even?
Honestly, I don't think that there's anything online or books that will prepare you on a journey. I think the best thing is to do an internship in a lab, which is how I started my journey with structural bio, because you can read all you want, but you will not be prepared for the amount of failures and how good the success feels in this field. And a lot of professors are very willing to get a student and to sort of train a new wave of scientists. So if you're thinking about protein crystallography, then I imagine you're at university. And if you're at uni, you should be able to get an internship with a prof. That's my thinking.
Yeah, so we're looking for students as well. Apply. You can make a summer internship, and you can either go to the lab and try to make crystals, or you can go to the beamlines that right around the corner and measure data, or you can sit in an office and program some things.
So
and I'll teach you. I even had a course last year where I taught people.
All right.
Keith, do you have any resources or books maybe?
Other than the holy sacred text of the Principles of Biochemistry, Leninger and Koch, 7th edition, then I don't. Other than that, I think you're fine. I think what Michael said is probably the most sensible and most pragmatic, especially if you really want to get hands down and deep. People could tell you how to build an airplane, but you won't know how to do it until you do it. So yeah, I would recommend just getting into it. And there's plenty of people who are willing to take students who are eager and willing to learn and understand.
If there's a book that I were to recommend to people willing to sort of learn a bit about crystallization, crystallography, but not brilliant with maths like myself, one good thing is Crystallography Made Crystal Clear. It's a book that was sort of my savior, and it explains it's good when it comes to putting you to sleep, especially later on, but it does explain things in a relatively clear, non-too mathematical manner.
Ooh, we've been going a while. Thanks, everyone, for sticking around and doing this very interesting. Probably got one more question, unless all of you got any pressing questions, but no.
There's some in the chat, though.
There's always more questions. But maybe the last question, I think, because you mentioned that some people are saying that X-ray crystallography is kind of, and protein crystallography is kind of, dying field. I mean, that's a very easy thing to throw out, and then, every 10 years say, oh, this field is dying, this field is dying. But say this video, lasts on YouTube until the end of civilization in 3 years or whatever. Where do you predict the field is going? Do you still think it's going to be the same? What do you think will change in 10 years or so? Say if someone's watching this now and they're in high school and they're going to be, doing PhD in, 7 years or so, what do you think will change, or do you think it will be sort of the same?
I think this question is best directed at Wolfgang to answer first.
Yeah. Is it just about improving the technology, and it's just sort of the same techniques? It's just going to be faster, better, harder, stronger with better technology? It's ending on a tough question.
No, it's a good question, and I think it will, for a long time, be the method of choice. Just if for pharmaceutical companies to find a structure, I think this will just stay the same for a long time. But what we want to do is more than just a structure. We want to see how molecules act. So that the vision is to have a movie of a protein in action at some point. And I think we have 1 or 2 cases where we can actually see how a protein crystal has changes within a small range of time. For example, if you imagine a photosensitive protein and you crystallize that, and then you put it in a beam and then you hit it with a laser, then it reacts to this excitement, and it does some structural changes, and you can see these structural changes and go along as it undergoes these changes and then comes back around to the original ground state. And this is very exciting, and this is what we want to do. Most of the cases, it doesn't work like that, but when it does, it's especially nice. But then, of course, we are under a lot of pressure from electron microscopy, because electrons interact more strongly with proteins. But then, on the other hand, in order to get very nice data from electron microscopy, you also need to make very pure, very stable, very rigid proteins. And then at that point, you can basically already make a crystal out of it. You can make a crystal, or you can put it into the electron microscope. It doesn't really matter anymore. So even if you learn how to purify a crystal, and then at some point it turns out that crystallography is dead, then your know-how is not wasted. You can just make the same thing and put it in the electron microscope. And then another thing is, because electrons interact more with proteins, well, why not use electrons to do crystallography? And electron crystallography, I think, is going to come, especially because you've seen these synchrotrons. They are large, they are expensive things. They are millions, if not, well, not billions, but they are expensive. They are millions, many millions expensive. But an electron microscope is not that expensive. And an electron microscope that only does diffraction is even cheaper than an electron microscope that does, because if you want to do electron microscopy, you diffract or you interact the electrons, and then you refocus to get an image. But you don't need to do that if you just diffract. So it's even cheaper, and you can build it for a few tens of thousands, basically. And that's what we're also doing. So we want to explore all the things. Of course, not all of the things. Actually, most of the things that we tried don't work, but but we're still trying. And so this is one other thing that will probably come, is that you do crystallography with electrons.
I mean,
Heath, any bold predictions? Oh, no, okay. Yeah, no, you go. Bold predictions, yeah.
My prediction is that, so as Keith has mentioned before, like essentially, if proteins were to sample every conformation as to go to the right fold, that would take longer than the length of the universe, essentially. So there is enough information within a sequence of the protein to sort of direct you towards what will be the correct conformation that protein will take. So right now it's of course proven to be too difficult to predict that with our current technology, but everyone's excited about quantum computing and it being able to solve really complex problems such as hacking, or in this case, it could be solving the structure of protein just based on its sequence. So I imagine that maybe within, God knows, definitely decades, if not longer, we should hopefully be able to then just, input a sequence of a protein into a program, and hopefully it will be able to spit out information on the probability that this protein will take this conformation. And well, that will make all of us out of our jobs. But how tempting of a concept that will be.
Mm-hmm. I mean, you can't, the issue with people saying that a field is dead is because they have no idea about how the field will evolve and the new technology, because they never account for that. And they just assume that nothing changes in the field and everything stays as it is. The
structural biologists will always be needed. But the technique which we'll be using will be changing. X-ray crystallography is decades and decades and decades old. And there have been huge developments in the field, such as better luminosity of the beam, better detectors, cryoprotection. But the technique might change, but structural biologists will always be there.
Keith, what's your prediction for the future of the field?
I think in terms of structural biology, I foresee in the future being able to track the individual little movements of individual reaction mechanisms and so on and so forth. So
that'd be amazing.
If you were able to somehow crystallize any kind of protein through a surrogate crystal or some other mechanism, just in order to orientate these molecules in a way where they can be diffracted, that could be a possible another way to get over the crystallization bump, whether or not it crystallizes. And cryo-EM is not nearly as refined as crystallography is, and that still has a very long way to go. So I foresee maybe hopefully a potential marriage between the two, where you could be able to both see these larger molecules at very high atomic resolution. And of course, you do have plenty of structures of cryo-EM that are able to get very good resolution of larger molecules. So, yeah.
If I may also, I see a question, someone's asking how easy it is to determine the structure of protein that's bound to a substrate or transition state. Transition state will be hella difficult.
Yeah.
But solving a substrate bound or a ligand bound structure is something that we do routinely. Sometimes it can be difficult because just because of the chemistry of substrate. If, for example, your enzyme catalyzes an ester bond and your substrate is an ester, then it's going to be very difficult to capture those two interacting together. You'd have to create mutants, which are catalytically dead, which is what I've been talking about in my earlier presentation. So yeah, it's definitely doable. It's just sometimes it's a bit more difficult than other times.
Thank you so much, guys.
Yeah. No, thanks a lot for you guys all messaged the Twitter account, which I'm once again shouting out people to do. And there's a lot of researchers on Twitter, and I'm sorry for the people I also harassed this episode to come on the show. But I will continue to do that. So if you're a researcher, expect to be harassed. So thank you very much. I suppose we'll leave it there, and it's been very interesting.
Thanks for having us.
It was really cool. Thank you. And thanks for the people watching and all your questions.
Oh, yeah, of course, chat. I don't know how to end this. All right.
Care, nerds. Later.
Bye. Thank you, everyone.
Bye.
Next episode should be an organic chemistry theme.
Maybe.
Probably.